The GU Lab at the University of Washington is developing a next-generation biochemical tool to analyze protein interactions. This technology is expected to fundamentally speed up protein functional analysis and engineering. In nature, most proteins form complexes to play biological functions. Analysis of protein-protein interactions is fundamental to the life sciences and protein engineering. For many years, scientists have dreamed of directly analyzing all types of binary and higher-order protein interactions without separating the proteins from a complex mixture. However, all existing technologies have no such capability. So what are the challenges? To do this, we must address three major challenges. First, sensitivity. We need to count free single-molecule proteins and protein complexes. This will be possible if we can amplify single-molecule signals. Second, multiplexity. To analyze many, for example, thousands of different proteins at once, we need to identify each of them in a single mixture. Third, in situ detection. By this we mean we need a way to see which proteins actually form complexes. At the GU Lab, we will develop an in situ protein interaction sequencing technology to solve these problems. Let's take a closer look at the technology. Our first step to detect protein-protein interactions is to attach a unique DNA identifier to each protein subunit, called a barcode. The barcode will serve two functions. First, it will be used to amplify single-molecule signals. Second, it will be decoded by DNA sequencing to identify the protein. In principle, we can use DNA to barcode an unlimited number of proteins. The barcoding sequence can be actual protein coding sequences or program synthetic sequences. There are many barcoding methods. Let's take a look at two approaches. The first method is called ribosome display. In ribosome display, we start with the mRNA containing the gene of the protein of interest. Reverse transcription generates the cDNA barcode. The ribosome then translates the protein in a cell-free system, and then stalls in the absence of a stop codon, forming a protein cDNA barcode complex. This method can barcode up to hundreds of millions of different proteins in a single assay. Ribosome display is limited to proteins labeled with DNAs, so it can be very useful for protein engineering. For unlabeled proteins from biological samples, we need an alternative method for the barcoding. We can indirectly barcode proteins by using barcoded binders that recognize proteins of interest. For example, we are using nanobodies, which are small antibodies that contain a specific region that binds to a protein of interest. The nanobody can be coupled to a barcoding DNA via an enzyme tag. Our lab is developing a technology to simultaneously select a large set of nanobodies targeting many proteins at an affordable cost. Using either the ribosome display or the nanobody-based method, a specific DNA barcode will be attached to a specific protein of interest, from here on shown in this simplified representation. A library of barcoded proteins are then immobilized into an ultra-thin layer of hydrogel by mixing a gel precursor and two DNA primers. The gel mix containing proteins is dropped onto a round cover slip and then spin-coated to a gel layer of a few microns thick. Next, the cover slip is assembled into a flow cell. Let's take a look at what will happen next inside of the gel. The first step is barcode in situ amplification. Recall that each subunit of the protein complex bears its own unique DNA barcode, which are all anchored to the gel. The gel also contains immobilized forward and reverse primers for amplification of the barcoding DNAs. In a process known as bridge amplification, the barcode DNA is first denatured to a single strand. Then the free end of the DNA anneals to one of the primers. An extension step then makes it a double-stranded DNA. The same process repeats itself with other primers present in the gel until a very high copy number is achieved. These DNA clusters, known as polonies or polymerase colonies, can get amplified to greater than 100,000 DNA molecules per cluster, providing bright fluorescent signal for sequencing. The size of these clusters is typically around 1 micron in diameter. In the case of multiple barcodes from a protein complex, they will be amplified into spatially colocalized clusters. Now that we have sufficiently intense signal from amplification, the second step is to sequence the barcode. Let's use a single barcoding DNA as an example. The double-stranded DNA is cleaved and denatured to a single strand for sequencing, which will serve as the sequencing template. A sequencing primer hybridizes to a universal sequence on the barcode. A mix of polymerase and fluorescent terminator nucleotides are added. The complementary base is incorporated, then emits a fluorescent signal, which is captured by a CCD camera on the microscope. Next, the terminator is cleaved, allowing the next base to be incorporated. This process repeats itself until the whole barcode is sequenced. Sequencing simultaneously occurs in all single-molecule DNA copies from the same cluster. For example, here the barcode 1 cluster. Different barcode clusters corresponding to the other subunits 2 and then 3 are then sequenced one by one. Spatial colocalization of the barcode clusters indicates protein-protein interaction. A large number of clusters, some single and some colocalized, are imaged in parallel. The occurrence of the colocalizations are counted and subjected to statistical analysis to measure protein interactions. The fluorescent sequencing signals are recorded by scanning the entire flow cell. Here's an example of 3 sequencing cycles. Our current flow cell setting will allow us to analyze more than tens of millions of single or co-localized polonies in each experiment. So what are the advantages of our method? Let's take a look at a simplified example. Assume we have 3 protein libraries, each containing 100 proteins. The proteins can all form trimeric complexes with proteins from the other libraries, giving 1 million possible protein complexes. Our technology will be the only way to test these combinations in a single assay. In addition to protein-protein interaction analysis, our technology can be further extended to analyze protein nucleic acid and protein small molecule interactions. In situ sequencing can be performed in 2-dimensional or 3-dimensional structures, such as single cells and tissue sections, to analyze protein interactions in their native environment. Finally, we can apply our technology to engineer protein interactions to make protein sensors and switches, map cellular interactome networks, or screen for drugs targeting protein interactions.